Prokaryotes and eukaryotes regulate groups of genes at a time. Eukaryotes typically do that through the influence of the same transcription factors, whereas prokaryotes use operons, and we'll look at the lac operon as an inducible system. So again, when we start getting into gene expression, it's not so simple as DNA makes messenger RNA makes protein. We get into these regulatory sequences. So we have our genes, our DNA, but we also have these other sequences that are going to influence how genes are turned on or held off. So prokaryotes, if you see an operator, that's going to be thinking prokaryote bacteria. If you see enhancer regions, we're typically thinking eukaryotes. So again, to start out with eukaryotes, when we talk about coordinated regulation, a lot of times it's having multiple genes, all having similar transcription factors that bind to the promoter regions to start these genes together. So again, as we go through this whole expansion of gene expression in eukaryotes, activators bind to enhancers, transcription factors bind to promoter regions, and RNA polymerase then can bind onto the promoter region with the help of those transcription factors. Now, the DNA folding protein brings the activators over to the promoter region so that RNA polymerase can actually read the gene. And now we're looking at this specific p53 gene, which is a tumor suppressor gene. So as RNA polymerase continues and starts doing transcription, we start making messenger RNA. And that messenger RNA will grow, and ultimately we're going to have RNA processing. I'm leaving out some steps, right? It's a eukaryote. We have RNA processing. It leaves the nucleus, finds the ribosome. But here we are at the ribosome. We have that small subunit binding the large subunit. Again, I don't have transfer RNA, but as that messenger RNA is read, we're going to join together amino acids. And we have our primary structure of this long polypeptide chain, which again can go into secondary and tertiary structure. It becomes functional when it gets into that three-dimensional shape. So this from the p53 gene is making the p53 protein, which happens to be a transcription factor. That's going to hook onto multiple promoters. So not only by turning on one gene, we ultimately coordinate regulation and turn on multiple genes. So again, this transcription factor can help RNA polymerase bind to each one of these, and ultimately the tumor suppressor gene begins, and it helps with cell cycle arrest, DNA repair, and possibly apoptosis if that's needed. But all of these genes can be coordinated through a single transcription factor. Now, again, both prokaryotes and eukaryotes can have repressors involved. They are going to operate differently, but when we just focus on the prokaryote, it's going to be involved with the operator. But the formation of this repressor, again, follows that similar pattern where RNA polymerase is going to bind to the promoter region. It's going to make the messenger RNA. The messenger RNA is going to bind to the ribosome, which is going to join the amino acids to ultimately make the polypeptide and make the three-dimensional protein. So now we have a repressor, and that repressor can hold off transcription. So what we're going to look at now is this example of an inducible operon with prokaryotes. So in this case, the repressor is going to bind to the operator region. RNA polymerase is going to always bind to the promoter region, but if the repressor is here, we would consider this gene off because the repressor is going to hold back transcription. Now you can see down here there is no operator region, so this is always on. There's no place for the repressor to bind in this prokaryotic gene expression. But up here, the RNA polymerase is going to run into that repressor, and that is going to be off. Now this RNA polymerase can continue on, but I'm going to pause this so we can focus up here. So this inducible operon, you need some type of chemical inducer. So an inducer is something that starts something or promotes it. So this inducer is going to come and bind onto the repressor protein. It will remove it from the operator, which will allow the gene to be turned on. And when we say it's turned on, it just means that RNA polymerase can now progress and begin transcription. So as it does transcription, we get messenger RNA. Messenger RNA finds the ribosome, and we start building amino acids together, and we get a protein. Now, in this case, this protein is going to be, in its three-dimensional shape, an enzyme. And this enzyme is then going and breaking down the inducer. So, again, I'm pausing up here now to look back down here. Again, this is on, so this is always going to be working. And it's going to make that messenger RNA, which would go to that ribosome. But now I'm going to pause this, head back up here, and again, this enzyme is going to break down the inducer, break down the inducer, break down the inducer, until there's no inducer around anymore. And this is now still continuing to build repressors with no more inducer, because the enzyme that was created from this gene broke down the inducer. The repressor can rebind to the operator and turn the gene back off. So the thing is, we only make these proteins from this gene when the inducer is present. Otherwise, we don't want to just be making proteins for the sake of making proteins. It's only in the presence of the inducer that we make the gene to interact with the inducer. And again, when it's off, it means that nothing can happen until the inducer shows up again, and then we'd start the process over. The big example that we're going to look at is the lac operon. And with operons, it's a group of structural genes. Typically, these carry out a coordinated response. So with eukaryotes, we had the transcription factor turning on multiple genes. A lot of times, these genes are just linked together. So once it's on, it'll make multiple genes and multiple proteins that together carry out a functional response. So the operator, again, is where the repressor binds. The promoter is where RNA polymerase binds. And we also have this regulatory gene again, and that's what's making the repressor proteins. So with the lac operon, we're talking about the bacteria that's making is E. coli. And not all E. coli is bad. Sometimes you have E. coli in your intestines that are actually preventing some pathogenic bacteria from taking hold. And a lot of the bacteria in our gut is super important. So with our lac operon, it involves lactose, milk sugar. So as we drink milk, we are ultimately feeding ourselves, but we're also feeding the bacteria that are symbiotically living in our guts. So with lactose, it is a disaccharide, and it needs to be broken down into monosaccharides to be used for cellular respiration. And in order to go through this hydrolysis, lactase enzyme is needed. So it's the lactase enzyme that breaks down the lactose sugar from a disaccharide to monosaccharides. So again, bacteria will transcribe the lactase gene and translate the lactase protein, but only when lactose is present. If lactose is not present, there's no need to make lactase. So, another time through this, again, when we look at that circular chromosome in the bacteria, there's going to be a sequence where there's promoter, operator, and genes. So it's going to be off when there's a repressor on the operator, because RNA polymerase is going to get blocked and cannot do transcription. When lactose is present, it will lift the repressor off of the operator, which then allows RNA polymerase to bind, to do transcription, and ultimately that messenger RNA will be translated into the lactase enzyme. That lactase enzyme will interact with lactose sugar and ultimately break it down into glucose and galactose. When it's all broken down and there is no lactose present anymore, the repressor will bind back to the operator, which stops the transcription of the lactase enzyme, of the lactase gene. Another version of looking at this, again, we have regulatory genes, we have promoter, operator, and genes. And again, it's multiple genes that are working together. So again, the regulatory gene makes the repressor. The repressor is going to bind to the operator, which will prevent RNA polymerase from binding and actually reading the gene. However, if lactose is present, it interacts with the repressor and makes it inactive, which allows RNA polymerase to bind. Again, this means it's not binding. It doesn't have the right shape or chemistry to bind anymore. And RNA polymerase can then do transcription, which will make the messenger RNA and ultimately make the enzymes through translation. So one last thing here. We talk about activators binding to enhancers in prokaryotes, but we can also have activators in eukaryotes, but we can also have activators in prokaryotes. So we have this cap protein here. Now, lactose may be present, but if glucose is present, we really don't need to break down lactose. So we already have glucose and we can break things down, but if glucose is scarce, what we're going to get is a lot of this cyclic AMP. Again, we think about ATP. This is AMP. It's a 1-phosphate hooked onto the adenine. So we have cyclic AMP, and what cyclic AMP can do, when glucose levels are low, we're going to have a lot more cyclic AMP, and that's going to hook on to this cap, which hooks onto this binding site, which just makes the RNA polymerase grab onto that promoter, and then this works much better. So again, prokaryotes and eukaryotes regulate groups of genes at a time. A lot of times in eukaryotes, it's through the same transcription factors, and in prokaryotes, that happens through operons, and we looked at the lac operon as an inducible system.